So, very much welcome to this event on brain imaging and brain imaging for improved research and therapies that we are organizing as part of the Health Talks event series. In today's events, we have experts in brain imaging and brain therapies to discuss new technologies based on software, artificial intelligence, and medical device hardware technologies also. This is being organized this time between Kuopio Health and Aalto University, supported by Terkko. I just mentioned Kuopio Health is an innovation ecosystem. It works to enhance well-being, nutrition and food industry, medtech knowledge, know-how and research, and promotes agility in networking and interactions between companies and the public sector and academia. I myself am from Aalto University. Aalto is an Espoo-based University of Science, Technology, Business and Arts. And within Aalto, health and well-being is one of the 7 key research areas of our university. And among our focuses, our largest focuses within health and well-being is medical devices, where probably the strongest focus is brain imaging actually. And in addition of that, we also have neuroscience research among others, and that's also relevant for our event here. Generally speaking, Health Talks is an event series on health, life science, and entrepreneurship topics, organized by many actors, including Kuopio Health, Aalto University, and others, centrally including Terkko Health Hub. I'm soon going to just hand over to our speakers. We have, I think, very good experts in brain imaging and brain therapies who will discuss important and interesting questions here. Our questions more have to do with specific imaging modalities such as magnetic resonance imaging, functional MRI, and also transcranial magnetic stimulation as a therapy technology. As a first speaker, we have Dr. Jaakko Paasonen. Jaakko has more than 10 years of experience in the development of functional magnetic resonance imaging technologies, and he has a PhD in imaging from the University of Eastern Finland. Jaakko is going to talk about a silent and artifact-free MRI method that will complement traditional methods significantly, since those methods tend to suffer from acoustic noise and movement artifacts. So, Jaakko, please, the floor is yours.
Thank you, Markus. So let me share my screen, and I would be happy if you then let me know if you can see the slides. So you should see them now, or can you see the slides?
Yes.
Good, thank you. So, if we think modern neuroscience, understanding the link between the brain activity and behavior is one of the key challenges, or has been one of the key challenges for decades or even centuries. For example, to understand the motivation, emotion, decision-making, social interaction related brain activity in healthy and diseased state would help to improve the diagnostics and therapies. However, there are no efficient tools in behavioral neuroscience to record whole brain activity during complex behavior. So there's several imaging modalities and electrical measurement approaches, but they all have their disadvantages regarding behavioral studies. For example, there's not enough spatial or temporal resolution to record the brain activity, or the subject is, or the approach doesn't allow the subject to express behavior or even have a conversation. So ideally, you would like to have a method that allows as natural environment as possible, but this is challenging for behavioral research. So when we talk about brain mapping, so functional magnetic resonance imaging, or fMRI, is perhaps the most common brain mapping tool. So many of you might be familiar with the MRI scanner, and It has several advantages, like it's non-invasive, doesn't induce any ionizing radiation, and it has good or at least decent image and time resolution. And when we measure brain activity with MRI or fMRI, it's actually measurement of local vascular changes that occur near the brain activation site. So just a brief introduction, we have this simplified illustration where we have a neuron astrocyte and small blood vessels in the brain. So we have the action potential, so neuronal activity. So that means that there's a need of oxygen and glucose in the neuron. So that leads to signaling to the blood vessels to induce a vascular response. So basically there's an increase in blood volume and flow and oxygenation and glucose. But the changes in volume flow and oxygenation are something that can be measured with fMRI. So it's an indirect measure of brain activity. And here you can see some of the early brain mapping results in human brains. So there's examples of motor, somatosensory, and auditory and visual activation after stimuli. But fMRI can also be used to measure resting state activity in addition to the stimulated activity. So fMRI is traditionally done with echoplanar imaging sequence. And the sequences can be considered as a program or instruction for the MRI scanner. And different sequences, they produce different images in the same scanner. So MRI is very versatile brain imaging tool. However, this EPI-based fMRI has several limitations. So first is the very loud acoustic noise, which can be over 130 decibels. So if we look this chart here, so it's comparable to jackhammer or even gunshot. So it's really noisy approach. In addition, as MRI generally, it's very sensitive to movement and image artifacts, and EPI sequence is particularly sensitive to these. And here's one result of a clinical study where they found out that almost 60% of all clinical data is affected by motion artifacts. And also EPI sequence is not very well compatible with sensitive electrical devices or simultaneous electrophysiological recordings due to the fastly switching gradients. So overall, EPI fMRI is very uncomfortable and restrictive environment when we think about behavioral studies. So to overcome some of these severe limitations, what I just discussed in traditional EPI fMRI, we introduced recently a novel zero ecotime fMRI approach, which is based on so-called MB sweep sequence. So it comes from multi-band sweep imaging with Fourier transformation. So I'm not going into more detail where it comes, but I just want to mention that it's fundamentally very different from traditional EPI sequence. And those who are familiar with MRI, so few technical notes, which might be interesting. So it's a three-dimensional radial acquisition with very high bandwidths in all three directions. And it has minimal gradient steps, very short repetition time, and practically zero echo time. So it's not sensitive to image distortions at all, and it doesn't require a careful shimming. And if we think about the fMRI, so the contrast for the brain activity comes from the mainly from the blood flow. So the original purpose why to implement something new for fMRI was to image regions that are not accessible with the traditional EPI. And in this example, I have two studies. So deep brain stimulation fMRI and spinal cord stimulation fMRI, both done in rats at 9.4 Tesla. So if we take a look first, the deep brain stimulation, which is used also in humans to treat some, for example, Parkinson's disease symptoms. So we have this electrode implanted in the rat brain, the thalamus. And in the bottom, we have the traditional EPI images, which show the activation to the stimulus. But basically around the electrode, we have this black hole. So 1/3 of the brain is gone because of the image distortions because of the electrode. But in the MB Swift image, so the novel approach, we just see this clean line for the electrode, and then we can see also the activation at the electrode tip and also at the projection sites in the cortex. And in this lower example, we have the spinal cord fMRI in rats. So this is the EPI image. It's very blurry, it's very distorted, and you barely can guess where the spinal cord is. So it should be here where the blue dashed line is. And these colored pixels tell where the activation to the spinal cord stimulation occurred. But if we look at the novel approach, we have a clean image with basically no distortions, and we can see clearly the activation in the spinal cord due to the stimulation. So overall, the novel method shows superior image quality compared to EPI in these challenging circumstances. From a more practical point of view, so we measured that our method is up to 32 decibels more quiet than the EPI. So it's really a significant improvement. So if EPI is comparable to gunshot, so MB Swift is comparable to restaurant environment. So it's not silent, but it's much better. And we have been working to improve the acoustic noise even more. So currently we can get quite close to the background level. So it's improving all the time. And importantly, MB Swift is not sensitive to body movement. So we had an experiment where we put an anesthetized rat into the scanner, and then we remotely moved the body of the rat. So blue line is the traditional EPI method, and when there's a movement, we can see clearly that the signal is distorted during the movement. But the novel MB Swift method doesn't really care about the movement at all. So it's really robust approach. And additionally, the novel method interferes only minimally with the electrophysiological recordings. So here's examples of EEG signals measured simultaneously with the MRI. So MB Swift is rather clean, even the MRI sequence has started, but in the EPI sequence, there's huge artifacts coming from the sequence. So it's very much more compatible with sensitive electronic devices and measurements. So as I've shown you, the novel method indeed overcomes many of the limitations of EPI. So it's quiet, insensitive to body movement, and insensitive to image distortion. So it's really promising. And we started to think that in theory, this method should be a very good approach for behavioral studies where the scanning or experiment is much more complex or demanding. So we wanted to try something new. So something that has not been possible with EPI, like even though this deep brain stimulation spinal cord is challenging with EPI, but something that really hasn't been able to be done with the EPI. So we trained rats to be rats for head fixed imaging in the MRI. So the rats were able to walk on the slippery glass and express spontaneous behavior. So their head was fixed through a head implant. And the rats spent 15 to 25 minutes in the 9.4 Tesla MRI. And during the measurements, the rats were mostly passive, but they also exhibited some spontaneous activity, like eye movement, limb movement, jaw movement, sniffing, whisking, and twitching. And we followed the spontaneous behavior through a video camera inside the MRI board. So first we performed a data-driven functional parcellation for the data. So to simplify for those who are not familiar with it, so basically this is done with independent component analysis, ICA. And these components on the right, they are thought to represent a structure of brain functional networks. So each component is like a distinct brain network because they share some kind of similar signal behavior. And we found out that what we got from these spontaneously behaving rats, so these components were really anatomically relevant and symmetrical indicating that the data quality is really good. And also these results were in line with previous studies in awake restrained rats done in other modalities. Then here are a few examples of activation patterns during the spontaneous behavior. So in the top, we have the limb movement. So or rat tried to run or walk on the slippery glass. So we got really nice and localized activation pattern in the somatosensory and motor cortices, which also is shown here in the video. Then another example is when the rats were grinding their teeth. So we saw activation in certain motor and stress-related networks. And third example is of sniffing. So then we saw activation in exploration-related regions. So these are just like a scarce results. If you're interested in details, you can find them in this very recent paper. And here I just wanted to show one individual event where we have the video of the rat inside the scanner. We have the brain slices here that show the increases in signal, and then we have the signal time courses from 2 different regions. So this is, let's see, yep. So this is related to whisking. So the rat starts to whisking now, so it starts to move with whiskers. And now we pause the video so we can see the signal increases in the barrel circuit of the rat in many regions. And we can see that the signal goes up in these barrel circuit regions, and then it slowly goes back down. So we want to conclude that we have demonstrated that novel methods like these MB-Swift fMRI, they enable for the first time whole brain behavioral studies in spontaneously behaving head-fixed rats. And this further allows novel study designs related to, for example, cognition, tasks, physical exercise, social interaction, and so forth. And importantly, similar approaches, zero echo time fMRI approaches, they can be used with humans. They can be implemented into the existing scanners, and they can immediately provide some freedom in the behavioral research in humans also. But of course, you might know that the physical restrictions in the bore, they restrict the available study designs because there's not much space for the subject to move inside the scanner. But fortunately, one of the advantages, which I already mentioned, of these novel methods is that they do not need a large homogeneous magnetic field. So this means that we could, in theory, decrease the scanner size drastically. So if in this kind of setup, the subject could already have a social interaction, use some devices, or express some kind of behavior. And especially if we turn or tilt the magnet 90 degrees so that the subject is actually sitting in a chair, or if there's even 2 of these devices, so we could actually study brain activity during social interaction. Or if we lift the magnet a little bit, it might be possible to put a subject on a treadmill and walking on a treadmill and explore a virtual reality while the brain scan is going on. So if you're interested in this technical development, so there's a very recent review in neuroimage. So the take-home messages of this presentation is that novel zero echo time fMRI sequences can provide significant improvements into the whole brain imaging. And the EPI has struggled more than 30 years of these issues, and here is one possible solution for those. And we propose a shift towards these more convenient, artifact-free and quiet sequences that have less restrictions in behavioral studies. And all of this to facilitate the understanding of underlying mechanisms of behavior in the brain, ultimately. So I want to thank my colleagues and funding sources, and of course the audience for your interest. So thank you.
Thank you, Jaakko, very much. There was one question in the chat, and feel free to write more. The question is that how can we gain access to procure CPD points?
I'm sorry, was the question?
I'm not sure how it relates, but the question is that how can we gain access to procure or acquire CPD points? Is that something that you know
yeah,
about?
I'm unfortunately I do not understand the question.
I don't think it was related to the talk as such, because the question came there really early.
Okay.
I think it's some practicality maybe related to this seminar.
Okay.
Hanna, do you have any points from yourself?
Yes, I would, if I can start. So
Sure.
I would really great talk and great results. I think you are too modest in the what you say that could be done with this kind of methodology. So as a clinician and a clinical researcher, I actually think that that kind of possibilities of first of all getting better signal to noise ratio. You showed, I think, got marvelous results already for like single responses from rats. I don't know how that goes with humans, but anyway, better signal to noise ratio and getting quiet environment would already be an excellent addition to the clinical fMRI studies we have. So we don't have to think about going to behavior. Behavior, of course, is excellent for that kind of what we all want to do from the research perspective. But from the clinical point of view, even getting better signal to noise ratio, which because the patients, they move in the scanner, so getting that restricting that would be an excellent, and the quieter sequences is actually also clinically really relevant, because we are mostly actually using clinical fMRI in acoustic studies. So when we are looking at children who are evaluated for brain surgery, for example, so these are the most typical clinical applications of fMRI. So we would need quiet environment for the stimulation. We would need quiet environment because of they are typically children, so they are kind of afraid of everything that's going on there. So there are many kind of clinical practicalities that you would help with this kind of new sequences. So behavior comes at some point, that's great, but also here I can see many good applications in clinical practice, and quite rapidly if these are implementable. So could you comment on that? How soon could we get this into human fMRI practice?
I completely agree everything with what you said. So as like a general like a change of workflow, we nowadays do everything with these modern techniques, because they're, as you said, they're silent and they do not care about the movement. So basically any measurement would benefit of those only of those. So you don't need to go like complex studies. So when to get these to human scanners? So we have currently started work in one collaboration project with the Kuopio University Hospital, where the aim is in the latter part of the project to start to implement this kind of sequences to human scanners. So of course we don't have human scanner ourselves, but we are really like interested to start a collaboration work to start to put it in the human field. And this is something that has started to gain how to say, interest in the last maybe 1-2 years. So there's actually many groups in preclinical fMRI who have started to implement these kind of sequences, and we hope that it could spread to human scanners as fast as possible. But we are, as you may understand, limited because we have only human scanners, and then it's a huge jump to human. We have only rat scanners, so it's a huge jump to human scanners because there's license issues and patent issues, and the manufacturers don't give the access to modify the sequences so easily. But we have people who have undergone the the appropriate courses to modify the commercial devices for these sequences. So the work is underway, but unfortunately it's quite slow.
Thank you. If there's any other questions, please just you can actually just start to ask it, or if you want, you can write in the chat that you would have a question. That's also possible.
While we are waiting for other questions, I have one more about the field strength. So does it have an effect? So you have been now having this 9.4 with the rats, but what about human 3T, or does it?
Yeah. That's a very good question. So because these sequences are so novel, so we actually don't know exactly what is the origin for the signal. So it's under investigation. So we have currently an NIH-funded project where we try to figure out where the contrast comes from. And one part of that project is to test different field strengths. And that way we can get an idea. So what is the contrast mechanism and how it should work in lower fields. But this is our colleagues in Minnesota have done already some preliminary tests in humans, and I think it was 3 Tesla, and the results were comparable to EPL. So I mean, it should be doable in humans. So we have some ground, like preliminary data already.
Thank you. If I'm right, there may not be further questions. But thank you, Hanna. Already now, I'd like to tell everybody that I asked if Professor Hanna Renvall can also join us, and we are very happy that she could. Hanna is a professor and medical doctor from Aalto University and the Helsinki University Hospital, and she leads the Biomag Laboratory, which is a common research institute of these organizations, and it can be very useful. I think that Hanna can comment from the viewpoints of a doctor and a neuroscientist. So thank you very much for coming. Now I think it would be a good time to go forward. We have next Professor Jussi Tohka, also from the University of Eastern Finland. And Jussi is a full professor of biomedical image analysis and head of that image analysis group at the AI Virtanen Institute at the University of Eastern Finland. And Jussi's research focuses on developing new methods to analyze imaging data and developing machine learning approaches for predicting the course of brain diseases. And Jussi is a researcher who has worked at several universities worldwide, and some of the software tools developed by Jussi and his group are already in quite wide use in medical imaging laboratories across the world, actually. Jussi is going to talk of the title, speech title, Machine Learning in Brain Imaging. And machine learning is making its way, as you know, to really many aspects of life, and that certainly is true about medical diagnosis and treatment approaches as well, and I would imagine can be possibly hugely beneficial for brain imaging also. And well, Jussi, I will give the floor to you, and please just go ahead.
Thank you, Markus. Thank you for the introductions. And now you should be seeing my screen, hopefully.
Yes.
Good. So thank you for the organizers for inviting me to speak in this very interesting seminar series. And thank you, Markus, for introductions. And I will give you overview in our research in machine learning in brain imaging. And I can move my slides. Yes. So background for this research is, of course, that brain disorders are massive both economical and individual burden, and I'll do maybe individual burden to the individual suffering from these diseases, and also their close ones is more important, but it's easy to understand how big problem we are talking about when we go to the economical side of things. And in 2010 already, it was estimated that the cost of brain disorders in Europe is 800 billion euros. That is quite massive sum, and it is cost of brain disorders is increasing all the time. To cope with this situation, early and more accurate diagnosis is of course essential. Also, we need tools to follow disease progression and study the effects of potential treatments. And also we need better understanding what goes wrong, since many of there is no kind of cure for many brain disorders. There are of course therapies and medications which relieve the symptoms but do not change the cause of disease. And my argument that for these points we need brain imaging, because brain imaging is really the only way of studying, looking at the living brain at the system level. And there are of course several brain imaging techniques providing complementary information. As you can see here, examples of some magnetic resonance imaging sequences and some also PET imaging with different radioligands. And as we saw in the last presentation, potentially these types of imaging modalities is increasing also. And changes in brain often precede visible symptoms of the disease. This is of course well-known fact in dementia, especially in Alzheimer's disease, where the changes in the brain start to happen maybe tens of years before actual symptoms or clinical diagnosis can be done. So this also suggests that early diagnosis and also the follow-up of the potential treatments would be possible using brain imaging, and it maybe is not so easy with any other technique. And the second argument that I will make is that we need machine learning to be to really use this brain imaging data for understanding brain disorders and better prediction of brain disorders at the level of individual. So we can use machine learning for several purposes in neuroimaging. Of course, most simple example is image classification, which is in its simple forms is for the diagnosis support. What I will talk about is a little bit different. But then we can also use machine learning for image segmentation. That is essential to get quantitative values, quantitate this brain imaging data. And also machine learning is finding its way to image reconstruction very very importantly, very recently, and image registration. And we mainly deal with image classification and image segmentation type problems, or have dealt in the past. And what is really needed for machine learning algorithm development in these two applications? So for image classification, you need a large annotated training set, image data, and diagnosis. I usually consider diagnosis in X years more interesting. I will come back to that. And for segmentation, we need image data and manual segmentations. There we don't need so huge number of images. So in this presentation, I will go to the more detail about our research in image classification. But before going that, I want to show this slide. So different machine learning tasks require quite different machine learning tools. So if we talk about small or wide data, tools are quite different compared to what we need when we talk about really big data. And for example, segmentation image segmentation is typical example of big data, because there the samples of interest are voxels, and we easily have millions of voxels in our training set. But in image classification, at least in neuroscience, we are mostly within this small or wide data categories. We at most have some thousands of participants, and but we can have millions of voxels, of course. So it can be also wide data. But I think really what is interesting in machine learning combined with this image classification application, that you don't have to combine labels, so diagnosis or whatever, and image data acquired at the same time. But you can take images that have been acquired in the past, and then look at what is the condition of these participants currently, and that gives you kind of a crystal ball into future that would be difficult to achieve without utilization of modern computational tools. Of course, one of the most studied example of this image classification framework is to study whether individuals suffering from mild cognitive impairment convert to dementia or Alzheimer's diagnosis. And this is really well studied topic currently, and not so interesting anymore. So, but this is a figure from our paper from 2015. But this is really interesting. And why should we limit ourselves to imaging data? We can also combine in this machine learning framework other types of data. For example, in this dementia type applications, usually there are behavioral data and cognitive tests made to these individuals. So combining them with the imaging data is indeed beneficial, as you can see here. It's beneficial in MCI to AD prediction, and it's also beneficial, mildly beneficial for very short time window healthy to MCI conversion prediction. If we increase time window to be longer in this healthy to MCI prediction, we can note that brain imaging becomes more and more important all the time, but we don't have anything published on that yet. So this is quite general framework. So you can utilize this for predicting any brain disease or any endpoint. So having data at year 0 and then following up on those subjects, we can see what happened to them after X years. That gives quite powerful predictive models that can be used then for early diagnosis or potentially to follow up or to select subjects for the clinical trials. And of course, this is not limited to classification. We can try, for example, to predict future cognitive scores using in this case quite multimodal data. And it's useful to really consider multimodal data in this case. And what is needed in more detail? So data. So there starts to be quite a number of relatively large imaging databases of different degrees of openness, and they start to contain long enough follow-up periods. This is an example table in my recent Academy of Finland funded primal project about the data that we are mainly going to be using in this primal project. And all of this data except the Vallecas sample is currently available for researchers with different degrees of difficulty in obtaining this data. Of course, we could also be using clinical data, so clinically acquired data, but we are not quite there yet. There are multiple technical and also legal difficulties in using actual clinical data, but this is very interesting direction to move into. I would like to stress that validation of these machine learning algorithms is very, very important, and it is important to do it well. And it is sometimes it is not easy to do it well, because sometimes these pipelines through which images go and are processed are rather complex, and you need to really understand what is happening to the images when you go through these pipelines to be able to produce unbiased evaluation. Also, also often there is a situation that that there is a strong hype about machine learning or artificial intelligence, and unrealistic expectation what these methods can offer, and this leads to situation where thorough objective assessment of the performance is a task under pressure. And there are few do's and don'ts in this recent tutorial paper that we published with Mark van Kils. In this presentation, I would like to highlight the last point. So a small sample size, and if you have tens or even hundreds of participants, then counting-based error estimation measures tend to have large variance. And this variance increases when the model complexity of machine learning model increases. And this is basically pretty unsolvable problem. So it is important to also report the variance, some variance measures of your error estimates. Okay, I'll end with this slide, and I'll want to end with the words of Trevor Hastie, who said that there are some powerful and exciting tools for making predictions from data, but they are not magic. You should be skeptical. They require good data and proper internal validation. Also, I would remind that in brain imaging, data is wide. So there can't be thousands or millions of variables, but only hundreds of samples, that means participants. So there are some technical interesting aspects of machine learning applied to brain imaging data, some of these which are somewhat specific and some of which are more general. One interesting point is, of course, variable and feature selection and importance that has received lots of attention recently, but I think there are still important blind spots to be studied, especially in technical direction and applications in brain imaging. Combining imaging data to other types of data, such as neuropsychological data, genetic lifestyle, it has been this has been, of course, done, but I don't still think that it has been done very well, taking into account of special properties of imaging data, and our own publications in listed here are methodologically pretty simple. There is also difficulty in combining data from different sites, even if you wouldn't have legal difficulties in that. So that data is heterogeneous, and there has recently been quite interesting approaches to that. Some of which are cited in our recent review paper by Valverde. And also, diagnoses are noisy, and neuropsychological data is noisy, and data is often imbalanced, and error estimates are noisy. So there needs to be solutions for these problems as well. And with this slide, I'll stop my presentations, and if there are any questions.
Thank you very much, Jussi. Extremely interesting, and obviously on topics which have extremely much of importance, potential use for patients, but also for healthcare systems and costs in many significant ways. So really interesting indeed. Indeed, if there are questions, we can now take some of them. There's one question right now in the chat, which is that how much generalizable, in your view, is the work that you have done here?
Yeah, if you speak, if I speak about my work and generalization to other cohorts of similar kind, I think that the models that we have used are relatively simple, so this should be quite adaptable also in other cohorts, especially if you can afford a little bit retraining. In general, we are not only ones in the world that is doing research like this, and these research tools start to make their way into the clinical practice, and they have received many, or some tools at least have received FDI and CE, not by us, but for example, a company called Combinostics in Finland is doing decision support systems based on these kind of data. So they have obtained approvals for using the tools for clinical use. So I think this points out that as a field, as a subfield of imaging neuroscience, we are moving into direction that is maybe clinically going to be useful also.
Thanks, and very interesting. And speaking about others in the world who are doing research in this important area, Hanna, am I right that you also have been engaged in some research regarding possibilities of predicting neurological outcomes that could be dementia or Alzheimer's disease? And you also have a hand up, so please.
Yes, we are working on very similar kind of question, but from the EEG, MEG point of view, so using different kind of data. So I disagree with you on the that you said that this is not interesting anymore, that MCI and machine learning is not interesting. So this is the part where I disagree with you, but otherwise I completely agree with you on everything that you said. So I also think that we don't have these tools in the clinic yet, and this is why, because we still have problems there. We don't know what is the best data to put in. You also mentioned the neuropsychological data, genes, other blood biomarkers. So these are important. So we don't really know yet what are the best things to put into the model. And another thing that also you mentioned is that the data, that our going from the research where things work nicely, we get nice predictions, then go into the clinical work where the people have different kind of diagnoses, every country has different kind of specifications, so then it's a mess. So we need lots of well-stratified data. Hopefully we could, like the legislation would help us share data within our country, but also with other countries more easily, because this is exactly what we would need in order to get this into the clinical practice someday.
I completely agree with Hanna that really the thing here is not anymore demonstrating in that we can do this. We definitely can do this if we have lots of resources and lots of data actually. That is the difficulty, and really this data is very heterogeneous, and I don't think that the MRI is worst modality in that that sense.
Okay, thank you. I think we have time for one question still. Caroline George, please.
Hello. I think your talk was really interesting. I have never heard of using artificial intelligence and the actual machines to predict certain clinical disorders that you might see in patients or even in certain research participants. I think it would be really great for intervention, which is, like you said, if you can notice the differences in the brain before the actual visible symptoms occur, I think it would save money in terms of healthcare costs, and then also save the person's life eventually. I did think it was interesting that you said it would be best to incorporate non-imaging data as well as imaging data into the machine learning. And I wanted to know, do you think that that kind of non-imaging and imaging data interaction would increase or decrease the error of margin in terms of the AI's accuracy in making predictions of diagnoses?
I think combining different kinds of data is a necessity to get well working predictions, because I don't know why it's actually so, but different data modalities typically predict different aspects of the brain, and especially combining structural MRI and neurocognition is relevant, because other ones is about structure, and another one is about function. So it's kind of easy to understand on a surface level, but it seems that especially when the prediction window increases, so more and more years you wait until you make the prediction, is then a combination of several data modalities seems to be very beneficial.
Thank you.
Thank you very much, and I'm going to now go forward to our next speaker. Since the time requires that, but usually there are comments in the chat. I think there is one question there. If you have time, if it's possible to answer briefly in writing, please do so if you feel that way. But we are now going to Dr. Victor Hugo Sousa from Aalto University. He's a research fellow at Professor Risto Ilmoniemi's group at Aalto University, and he develops technologies for personalized brain stimulation with neuroimaging data. Victor Sousa has created and validated several human and preclinical multi-coil transcranial magnetic stimulation coils technologies to study network brain functions. And his topic today is personalized network stimulation, combining neuroimaging with multi-channel transcranial magnetic stimulation. And here we talk about the therapy technology of TMS, transcranial magnetic stimulation, which is known to have a good potential for several neurological therapies and for the study of human brain too. But most of these technologies applications are being delivered only over one cortical area, which is a limitation, and that is why Victor will present the development of a novel multi-coil TMS device. So please, Victor.
Thank you, Markus, for the invitation and also for the kind introduction. Our group in Aalto, as you summarized pretty well already, our group in Aalto University is developing a new type of device to stimulate the networks of the brain. So that's the topic that I'm going to talk about today. Let me just share the screen. Yep. Okay. Yep. So transcranial magnetic stimulation, abbreviated as TMS, is a technique that we use to send a magnetic pulse through the scalp and with the electric field activate the neurons in the brain, in the cortical surface of the brain. So for example, if we apply a pulse over a certain area like the motor cortex, the neurons can be activated, depolarized, and this activation can propagate through the cortical spinal tract and go to the muscles. And we can measure this activity in the muscle, for instance, with electromyography. From that response, we can, for example, extract the delay between the stimulus and the onset of the response. That's called latency, and also the amplitude. This gives us a direct measure of the whole system that is between the stimulation site and the muscle being activated. A TMS experiment is usually performed with an operator holding a stimulation coil on top of the head, applying a pulse, and then we have a hand movement. Then we record a response. So like you can see here in this video. So now the operator is trying to find the target location that has the biggest response in the muscle. So far, so good. This can be used to, for example, find locations or representations of specific muscles from the hand, from the face, or from the arm, legs on the brain, and sort of map the location that these muscles are represented in the brain. And this is very useful, for instance, when a patient has a tumor and then you have to remove that tumor and leave some adjacent areas that need to be saved to minimize the impact later for the patient. TMS is also being widely used for the treatment of depression. Then it's targeted on more frontal areas, as shown here in the image, and has demonstrated quite big potential to treat some certain types of depression. But as we know very well, the brain is not a single, not all of the functions of the brain are not localized in single locations around it. So we have this big network of multiple areas that are interconnected. For example, here on the motor cortex, interconnected with different areas. And we have, for instance, the dorsolateral prefrontal cortex, which is related to mood control, also connected to several other regions. And very few of these treatments or these processes, they take into account this connectivity of the brain. And also, the conventional way of doing TMS requires too much manual work. That's, as you've seen in the video. And then this results in a limited accuracy that you can localize things. It's a pretty slow process. It depends a lot on the operator experience and judgment, and is blind to the state, to the brain state or the activity, the state of the activity of the brain. So then how can we explore the full potential of the TMS technology and kind of interact with these other mechanisms like the networks of the brain or the brain state? For that, we have been developing the multi-channel TMS, which is also named multi-locus, or just abbreviated as MTMS, which comprises of a few coils. So in that case, that of the video, I was showing just one coil, but here we have multiple coils, one on top of each other. And with that transducer, which is multiple coils, composed by multiple coils, we can control the stimulus millimeter by millimeter and degree by degree of orientation without moving it by the hand so we can control it from the computer. And these pulses can be delivered to different locations just within milliseconds, which is the time scale of the communication, of the neuronal communication. But how then multi-locus TMS works? We have one coil, and then this coil generates a certain stimulation field on the cortical surface. And then we have a different type of coil, which generates a different type of field, and a third coil, let's say. And then we combine all of the fields from each of these coils with a certain current that runs through them, and then we can change this location electronically. So we built this transducer, this prototype, and also built the power electronics to control that. And this is in the photo how it looks like. It's pretty big compared to the conventional device, but then you don't need to move it very much. Here it's been held by a robotic arm, and then when it's placed on a certain location, you can control the targeting by the computer. The algorithm for that is almost on a click on the brain basis. So you click on a certain location in the brain that you want to target, and then we compute the electric fields, the electric field distribution on that location, and then with that, and then we can change to another location by clicking in the brain, and that moves to that location without moving the coil. So I'm repeating that a lot because that's the key message of the talk. Then if we do that over different locations, we can measure the size of the motor response, and we will have for example, the yellow balls here represent where the index finger was, the thumb was actually the most active, or for instance, the little finger has a bit different distribution on the cortex. In collaboration with the University of Eastern Finland, specifically with Jaakko Paasonen that presented here, he's a big contributor to this work. We have been also developing a device, a multi-channel device to be used inside the MRI with small animals. So this is a schematic view of the setup that we built there in Kuopio. And we built and installed the cabinet for the stimulator inside the MRI shielded room, and the device can be controlled just next to the computer that also controls the MRI. And from our cabinet, we have the cables running to the coils that are placed inside the MRI. So this is a photo of the setup there in Kuopio. And here is highlighted, for instance, the cabinet that's fixed on the walls here in the corner, the optical fibers that go to the control room, the connection box that allows us to put the transducers that goes to the MRI. We needed to build specific coils to operate in the MRI, because with the high field, 9.4 Tesla that they have there, it's a pretty difficult environment. So when we give the pulse, usually the coils tend to break because the forces in the plates are pretty big. So we went through several iterations until we reached this one that has resisted up to a certain level that we can stimulate the brain's red. Now here is the transducer inside the case with the RF coil to record the fMRI signal, and the holder that we use to put the animal plus the MTMS coil and the fMRI coil also inside the magnet. And this would be potentially used with those MRI sequences that Jaakko presented already. Here's just a quick technical check that we did. We put the rat with the TMS coils just to see how much the presence of the TMS coils would affect the imaging. And in principle, there was not so big interference. And we also measured how much delay we had. How long did we have to wait after the TMS pulse to start the recording before the signal was disrupted, the MRI signal was disrupted. Then we saw that, okay, until a certain voltage, 375 in this case, we needed to wait 6 milliseconds, which is a reasonable time for us. A quick overview on our latest developments, which is the tractography guided neuronavigation and robotic guided MTMS. Used to take into account the anatomical connectivity between different brain regions, we have developed navigation software that monitors the location of the coil, the TMS coil, and computes the tractograms, so the streamlines that are related to that specific area that the TMS coil would be targeting. So with that, we would inform the operator that, okay, for example, now I came to the frontal part of the brain, and that region might be connected to other locations, for example, on the back of the brain and on the other side. So this can be a useful tool on guiding the network stimulation. And because our coils are extremely heavy, and so compared to the conventional coils, and we want to automate all of the procedure to minimize the errors that come from the operator experience and so on, we are developing also robotic control. In that case, we set a target on the scalp, and then we ask the robot to place the TMS coil there. If the subject moves, the robot can compensate the head movement. So you can follow here that the robot gives space. Also, this is one of the safety features. When the subject moves too much, the robot gives space for the subject and then goes back to the target. And then look at the screen on the left side. When it reaches the target and everything becomes green. So let's say we are on the spot that we want. Then we can deliver all the treatments and the TMS pulses that we want. Our main vision is to have a helmet-like device in the future, so that we can stimulate any location in the brain, because now we are limited to a small region. And this is part also of a project for commercializing these devices in the near future. So far, we named it as MTMS, just to relate it to technology. So our main goals then is to understand brain function, automate and personalize the brain stimulation, and provide the technology that would improve the diagnostics and the treatments of multiple brain disorders. This is a work of a huge number of people, and has been funded by many different agencies. So thank you to everybody also that contributed to the work. Thank you.
Thank you very much. Any questions to Victor or comments?
There are many in the chat.
Yeah, there's one from Matthias Bremer, for example. I can read that, and I'm not sure if you, Victor, answered parts of these after Matthias wrote the question. But anyway, I'll just read, and you can then answer those parts that are unanswered at least. Are you subjecting the brain to the same stimulus, or only moving the location of the stimulus? And is the stimulus always the same, or can it be different for different targets or different areas or the current?
That's a very interesting question. So we try to simulate, when we do the electronic movement, we try to mimic the manual movement that we do with the coil. But of course, because the brain is extremely, that has this folded geometry, so it's very uncertain in a way that each location that we have, like how the fields are distributed there, it's affected by many different factors. So we try to emulate, to mimic the hand movement when we do the electronic movement, but of course the distribution of the fields are different for each location. So that's, I think, if I missed something, just say, but I think that would be the answer, yeah.
Then a question from Teresa Elonia. Is it the same user interface software that you use for MTMS and MRI?
No, so we have our own interface for the MTMS, and then we kind of trigger the two systems, the MRI and the TMS, with an analogic signal. So they are separate systems, and we just create the link between them, but they are different interfaces.
Matthias also has a further question, which is, would you have to stimulate a reaction to the target always from the brain, or could you stimulate the target locally as well with the same result?
I don't think I understood the question. If you want...
Yeah, I guess you always stimulate as locally as is possible in a non-invasive manner.
So what would be a reaction to a target? I think that that term I maybe didn't understand.
And a follow on, could you stimulate the brain with a reactor agent? I guess it refers to chemicals which you are not using.
Yeah, we are not using any agents. Target as in, okay, so for example, if a person moves the hand and then we stimulate the brain, it might be something like that. So that when the brain reacts to the stimulation. So let me try to digest that. You have to stimulate the reaction to a target. Okay. Yeah, I don't know. Yeah, I don't know how the chemicals would be used in that case specifically. Yeah, and how would that be applied? So, but potentially yes. Let's say yes. But we usually stimulate the brain. So, for example, one possibility is that we stimulate the brain when the hand is doing a specific action. And then with that, we can pinpoint with more accuracy which kind of phase of the movement that we are interfering with. That we can do with the TMS. So using these reactions of the brain, or of the hand, for example, to kind of pinpoint the function that we are studying. That can be done. About the use of chemicals, that I really don't know of. Sorry.
So thanks from Matthias. Hanna, please.
Yes, great. This is also great stuff, and I'm also, of course, somewhat familiar with your research and development. And I think this the possibility of making TMS more personalized, it's really important, because I think TMS has not kind of I think it hasn't really established its full clinical role yet, and I think that's partly because of their not being able to okay, from a technical point of view, but also from the non-personalized point of view, it has not gained enough, and this probably would give those tools. So what do you think? What do you have your own ideas that where this would maybe be most of use in clinical practice? I have some ideas, but what do you think?
What do you mean? Which diseases or which conditions?
Exactly. What kind of clinical applications you see the front line?
Yeah, I see the front line of TMS in diagnostics, because the best I've seen TMS perform is on the direct interaction with the brain, in the sense that you probe and measure. So you give the pulse and you measure reactions, and then you can create these markers that you would like, and that's kind of in a certain way easier to create scientific based or well quantified measures that can give you that. So I really like TMS as a diagnostic tool, even though it has some limited applications and so on. So I'm in big favor of that part. Regarding the treatment, I think that we will open another level of understanding of how TMS can operate when we start synchronizing this with both the activity of the brain and also the functional or the anatomical localization. For that, I think diseases like I don't know, for example, Alzheimer, that, as you mentioned, that can be quite interesting to see, because it depends on many different areas of the brain, and also because it's a neurodegenerative disease, right? So then you can really affect how the signal propagates after you elicit that. So I think that could be very powerful as well.
Yes, I think that especially the possibility of looking at the tracts, so then you can basically you don't have to necessarily stimulate hippocampus or something, but you could stimulate the tracts and the kind of the circle, which is affected. So because of course you cannot reach every place necessarily with TMS, but you could reach the circuit in that sense. And I also think that from the diagnostic point of view, for example, traumatic brain injury is something that especially that the mild ones are such that they are really hard to diagnose nowadays. So getting a different kind of reaction, and for example, measuring with EEG at the same time and kind of learning of what is wrong would even tell us about what's going on in the brain. So it would also kind of help us to understand the changes in the brain. So not only make the diagnosis, but also to understand the underlying
the development also. Yeah, thank you.
Well, thank you very much also to you, Victor. These have been extremely interesting presentations, I think, about very important topics within this topic that we have today, brain imaging for improved research, and also such therapy technologies such as TMS that can work in combination with brain imaging. Still, I think we don't have so much time any longer, but there's a chat question, and maybe you can check that, Victor, if it's possible to answer those in the chat. And Jussi has also answered the question to him in the chat. Thanks also to Hanna, to you, and also for everyone for participating, and also for Kuopio Health and Maria Terttu for your help in organizing this. So I think it has been a very interesting session in this afternoon. Thank you very much to everybody, and bye.
Thank you.
Thank you. Bye.
Thank you.